Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about sort of a conclusionary comments regarding the process of meiosis. Meiosis, the thing is about meiosis, it's not only important because it produces sex cells and it produces haploid cells for sexual reproduction, but even more important than just procreation, what it does is during the process of during meiosis, there's some events that occur that create different gametes, even from the same organism. And by doing that, by creating a variety of gametes from the same organism, what biology is assuring is that when those gametes meet up from two different organisms, there's going to be a multitude of variation in the next generation. So in other words, there's some assurances that every time organisms reproduce, there's going to be diversity in the next generation. And that's really, really important because variation is critical for populations to survive, especially when environment is changing. So to that end, I want to emphasize these three processes right here. If you can see crossing over, which occurs during prophase between the homologous chromosomes. The event during anaphase 1 when the homologous chromosomes separate, they separate randomly. So you don't know if at one time all the paternal DNA is going to go to one side, all the maternal is going to go to the other, or some could go and others can go. And so this is random. So random independent assortment. And then the third is once you've created all these gametes, the fertilization is random. The chances of the same gamete fusing with the same gamete from the mother and the father is almost impossible. And so these three processes right here are going to create variation amongst the offspring. And so let me just kick around each one of these. And let me start off by going up here, and I want to talk to you about this crossing over event. And let me sort of just go right here and go, all right, here's a cell, and it's a diploid cell that's going to go under meiosis 1 and make 2 cells, and then meiosis 2 makes 4 cells, like there. And I'm going to sort of contrast what it would look like with and without crossing over. And so there's meiosis 1, here's meiosis 2 right there. And so let me sort of simplify things and say that, here's an organism with one father chromosome and one mother chromosome. So these are the homologous pairs. Say that let's say that when prophase 1 occurs and the homologous chromosome synapse in the form of the tetrad, there isn't any crossing over. And so, okay, no crossing over. So what happens is the homologous chromosomes separate. So there's one going there over to that side. Let me just continue with this color. So one goes over there, and then these separate down there. And then the same thing occurs over here. The red chromosome cruises over here. So there's a haploid cells right there, here, haploid, because there's no longer diploid. And then here's meiosis 2. So this is, let me just write it, meiosis 1, meiosis 2, same thing over here. And then look at this. Here's the haploid cells. But when I ask you, how many different gametes? And you're like, well, there's 4. I know there's 4, but how many different kinds? You're like, well, and the truth is these are the same, and these are the same, so there's really 2 kinds. Okay, it's fine. But in this case, when one chromosome gets really close to the other chromosome and they're dancing, they're coming together, sometimes there's a little physical exchange. Look at this, little physical exchange going on there. So in other words, a little bit of green comes over here and a little bit of red comes over here. And so now you've created a mosaic right here. And so when the homologous chromosomes separate, what you've got going is it's not a pure red, and it's got a little bit of a green shoe on it. It's got a little green piece, and this green guy has a little bit of red. You're like, yeah, I could see that. And then anaphase, they separate. But what's interesting about this, check it out. Over here, let me go here. What's happening is what you create is something very, very special. You've now created 4 gametes. Over here you created 4 gametes. But see, there's a pure red one and a red one with a little bit of green, and then green with a little bit of red, and then a green. You've created 4 different gametes. So this means that one person, when they're creating their sex cells, is going to create different ones each time. And so when a couple has children, the chances of the offspring being the same are extremely remote. So crossing over is a way to sort of mix it up, mix up the DNA from one parent to another. Now, it should be emphasized, and I'm going to mention it right now. If there's an exchange, and you're, say, for example, if you recall the analogy, if we're dancing and we exchange shoes, if we have the exact same shoes, it doesn't matter if we're swapping them. It only matters if the alleles are different between the homologous chromosomes that you'll get something different. That's important. And so let's kick this around. Let's kick it around. And so, for example, here, when you look at this diagram over here, and you can notice the homologous chromosomes coming together, there's a synapse, there's an exchange, the point of crossing over is chiasmata. Look at this, there's differences, and so when the homologous chromosomes separate during meiosis 1 and then in meiosis 2, you're going to get 4 different gametes. So what occurs is during anaphase, when the chromosomes separate, the chromosomes are not pure, but rather they're mosaic, and so you get some differences. So the crossing over creates these recombinant chromosomes, is what I'm saying, which creates variation. And so let me come back over here and show you something here. So pretty cool animation on this. And so here's some homologous pairs. Let's let this sort of play out and animate it. So what, again, to emphasize why crossing over occurs is because that's a difference in mitosis and meiosis. During meiosis, the homologous chromosomes actually come together, and when they come together and find each other, there's that possibility that there can be a breakage. And if there's a breakage between, see how these are sister chromatids? These two are non-sister chromatids. So there could be an exchange between non-sister chromatids. And if that occurs, this is the point of crossing over. This black chromosome is going to have a little bit of red, and this red one's going to have a little bit of black. And so when these separate, you're going to get gametes that are 4 different. This one, and then that one, and that one. So you get some variation occurring. So that's pretty neat. Let's consider this. I like this right here because what it's getting at is, let me make it a little bit larger so you can see. What it's getting at is the fact that here you're putting in actual alleles here. Do you notice here it's capital A? Notice here on this chromosome, these are the same. Of course they're the same. These are sister chromatids. But between the pair, notice the alleles are different. And so now if there's an exchange, there's going to be something that's changing. In other words, a little swapping occurring. And so crossing over occurs during meiosis, and it recombines the genes. Sometimes it happens, sometimes it doesn't happen. But it occurs during prophase 1 because that's when the chromosomes come together. And so when they come together forming the tetrad and they synapse like this, what can occur is non-sister chromatids can actually sort of physically move over each other. Like, notice how this is going to happen. And so it's emphasizing the fact that those are 2 different. So when they come, well, here's something. So if they didn't cross over, so there's meiosis 1 right there, and then now here's meiosis 2, and then how many different gametes do you have? You're like, oh, I've got this kind and this kind. I know I have 4, but there's only 2 different gametes. So that's without crossing over. So if there was crossing over, there we go, oh, a little crossing over there, and then what happens is cytokinesis, 2 haploid cells, meiosis 2, sister chromatids separate, and ho-ho, look at this. Now we have 4 different kinds of gametes. So crossing over produces variation in your gametes. So that's pretty significant. And so let's come back over here and see what we can do with this. And so meiosis is going to do this. It's going to be able to allow crossing over to occur. So that's number 1 in terms of creating variation in your gametes. Another huge way in which variation occurs in the gametes is that we're dealing with a lot of chromosomes here. Like here's an example where there's 2 sets. So it's a diploid cell because there's homologous pairs, and there's 4, so the diploid number is 4. So check this out. One possibility during anaphase is that the 2 blue chromosomes go to one cell and the 2 reds go to the other one. That's a possibility. And then look at that. So you've got this kind of gamete and this kind of gamete. That's one possibility. But it's random the way the chromosomes get segregated during anaphase 1. It's called independent assortment. So you have to consider it. Like what happens if this pair, the big blue went over there and the big red went over there? You can't have one of the same. So if the big one went to one side, the other big one has to go there. So the small one goes there, small one goes there. But notice this. This is a different possibility than this. So independent assortment will create variation in offspring as well. And so it all has to do with how they line up and how the spindle fibers sort of pull them apart during anaphase. And so, the big deal of that is, remember that what's happening is if one chromosome has its kinetochores going like this and the other chromosome happens to be sort of just randomly going like this, then one's going to get pulled this way, the other one's going to get pulled that way. And so when there's more than one pair, you can get a variation. And so I hope this is helpful to understand this process. So in one scenario, the blue over there, red over there. There you go. Over here, it could be blue, red, red, blue. And so you get different possibilities. And so mathematically speaking, if you're talking about a set of 2 chromosomes, there's really 2 to the end, whereas n is the haploid number. So in other words, in this case, it would be 2 to the 2, so you get 4 possibilities coming from this. Although you could really step your game up if you're talking about human meiosis, because our n number is 23. So there's 2 pairs of 23. So if you think about it in all the different ways, one scenario would be if all the father went to the left and all the mother went to the right. But then it could be another scenario would be almost all the father but one mother, and then almost all the mother and one father. And so if you did the math on that, it's 2 to the 23. So there's about 8 million different ways in which the chromosomes can independently assort. I mean, that's out of hand. And so I think it's kind of neat. I got to show you just to drive this point home. It's a really important concept too. So check this out. Here's a scenario where there's just 3 pairs. Okay. So how many possibilities can you have for 3 pairs? Let me just throw that out. 3 pairs. How many different gametes or different ways can they segregate? Let's see if you can figure this out. So one way would be that they line up like this in anaphase, and then maybe purple and turquoise go this way. That's one scenario. Okay? And so there you go. And so the number of possible orientations. So it's 2, number of chromosome pairs, 2, and then it's 3 because the haploid number would be 3, so it's 2 to the 3. There's then 8 different possibilities of these 8 different possible gametes that can be formed just because the chromosomes segregate independently. Isn't that neat? So that's why it's 2 to the 23rd, or over 8 million. And so this is not even considering the fact that these chromosomes are not really pure, because as a result of crossing over, some of these are mosaics. And sometimes there's crossing over, sometimes there's not crossing over. So I know that that can get a little bit busy. Let me see if I can find myself back. Sorry, find myself back to the notes here. And I know that can get a little bit confusing, but this is my point to try to illuminate it. Is, you get these different possibilities, these different scenarios, but then when you consider the possibility of crossing over, it's even further. And then this is the third and final one, random fertilization. So in other words, you've got all of these different kinds. So in other words, any sperm can fuse with any egg, and who knows which one is going to be getting together with the egg. So the zygote, produced by the mating of the same two parents, the ova has an 8 million possible difference. The sperm is 8 million, but when you consider 8 million and 8 million, the result is 1 in 7 trillion possible combinations. So this is the same two parents. The chance of having identical twins is 1 in 7 trillion, 70 trillion, 1 in 70 trillion. And then when you consider crossing over adds more to this, it's basically almost infinite. And so the three ways in which, just to drive this home, sexual reproduction, or the production of gametes, creates variation is crossing over, random assortment of chromosomes, and random fertilization. Now, all of this wouldn't matter at all if it wasn't for the fact that the two chromosomes may have different alleles. So it doesn't matter if you're swapping the same sweater, but it only matters if you're swapping different sweaters. And so how do new alleles, how do new nitrogen-based sequences arise? It's mutation. And that can occur spontaneously. You know how this goes. It could be replication errors or something. But as a result of mutation, there's variation, and sexual reproduction mixes it. So it's sort of like mutations creates different playing cards, and meiosis and sexual reproduction shuffles them together, if you like that analogy. Maybe, maybe not. And so I want to talk to you a little bit about flowers, and it may seem a little bit like random that I would, but we've been talking about humans and that meiosis occurs in ovaries and testes, but there's other organisms that sexually reproduce. The flower is the sexual organ of plants that produce them, angiosperms. Let me introduce you to some simple flower structure. Most people are familiar with the beautiful petals, and these are very bright and conspicuous, and the purpose of them are to attract pollinators. There's male structures on a flower. They're called stamen. I find that to be kind of funny because the name men right there. So these are the male structures. And this structure right on the top here is an anther. This is where pollen is produced. And so pollen happens to be, let's see if I can write that. Pollen happens to be the male gamete, or the sperm. Right there, and I'll just put some dots right there for the pollen that comes off. You might be familiar with this. And it's like, well, how do they get up like that? Well, there's a filament that's holding them up. So a filament holds the anther up, and collectively these are the stamen, and there's many of them. The female, on the other hand, the female is called the carpel. And the carpel is made up of this one structure here. And it's a sticky stigma held up by a style, and then there's enlarged structures called the ovary. And inside the ovary, there's this little smaller structure, sometimes there's more than one, called an ovule. So this is where female meiosis is occurring. So this is where the egg is being produced right there. and this is where the sperm is being produced here. So what happens? You might know this. Wind could blow and the pollen goes like this. It's called pollination, and it sticks here, or self-pollination, since it's the same plant. The sperm travels down the style and it goes over here and it goes in here and it fertilizes the egg, and that becomes the zygote. That's a diploid cell, and then that will divide through mitosis, and that will form, ready for this, an embryo plant. Embryo plant. And that embryo will eventually be enclosed by the structure around it, and it will form a seed. And so let me see if I can emphasize that. So the embryo is inside of a seed. How do you like this? Every time you eat a seed, you're eating a baby embryo. And then, ready for this, the ovary that surrounds it becomes the fruit. So if this is a pea plant, let me just get crazy with this. Now this is obviously green, I'm drawing it in red. This would be the fruit or the pod, and there would be many seeds inside of it. But this could very well be an apple, or this could be an orange with the seeds in it. And so what I'm saying is fruit comes from flowers, and inside the fruit are seeds. I hope that's not new to you. So in this case, check this out. Here's a flower that we were just talking about. Here's a real one, real petals. This happens to be a purple petal of a pea plant. And so what's interesting about this is this purple pea plant, curiously enough, can produce pollen. You're like, okay, the pollen, I'll just go this, is the gamete. So it's pollen. And so inside the pollen, this is a haploid cell. So you would think, for the trait of petal color, you can say, well, it's purple. And so what's interesting about this is it does contain purple. Let me just put a P right there. And so on the chromosome itself, like this, you can say that there's something a segment of DNA that controls the petal color of purple. Okay? And so when this is being formed, when pollen is being formed, let's keep this in mind, that what we're really talking about is a diploid cell. I'm going to stay with one color. So it's purple. This is purple. And so meiosis goes like this. It forms two cells like that, and then it forms four like this, each with a chromatid in it, and it's purple, 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 and purple. So this would be the pollen right there of the purple plant, the pollen. You're like, okay, that's pretty cool. And then let's check this out. If you had a white one right here, you might think, well, that's interesting, that this plant is producing probably a white-colored petal, and it might be producing white because its pollen has a gene for white in it. It's only normal to think that. But what's interesting, if you've ever had an opportunity, and we'll talk more about this later, let me assure you, if you've ever had an opportunity to cross-pollinate a plant, like check this out. If you're able to like mate these two, so this plant right here is producing pollen right there, and this one right here, let's exaggerate the carpel here. And so what's going to happen is this pollen is going to come on over, it's going to attach here, go down, and then what's going to form over here is, let's keep it a little bit more real, a big pea pod like this. And inside the pea pod are some peas. And so what I'm going to say, I'm not going to go into the story very much more, but I'll say this. What's interesting about this is when you cross plants together like this, the pollen is the gamete, the egg is the gamete, the embryo is inside these seeds. This is the fruit or the pod. And what's interesting is like what will these plants look like? This is my question. Will they look purple? Will they look white? Because they're the cross between them. It's sort of a hybrid. These are hybrid. Will they look purple? Will they look white? Will they look something in between? Is it kind of a blending idea? Will they look, I don't know, what's pinkish? I don't know, maybe some of them, 3 of them will be purple, 3 of them will be white. I have no idea. Actually, I do. But this is a good question. This is what people want to know. How does heredity work? And there's going to be a connection between what we've learned about meiosis and patterns of heredity. And so I hope this is intriguing you. You might recall this somewhere in your past, but the way I'm presenting it, I hope it seems fresh. So I want to know what happens when you fuse 2 different gametes from 2 different plants that have different colors. What's going to happen? Is there any way to know, or is it completely random? We shall see. I hope you enjoyed watching this video on genetic variation, crossing over, independent assortment, and random fertilization. Thanks.